So our next guest speaker is Dr. Hugo Volkaert. He had a bachelor's degree and master's degree in agricultural engineering in Belgium and got his PhD in forest resources from University of Maine, the United States of America. Now he is working with the Center for Agriculture Biotechnology in Kasetsart University, Thailand. His research of interest are in population genetic diversity and phylogeography of forest trees, genetic diversity and domestication of bananas, and also in some economics plants such as the oil palm. Not only the diversity, he also used biotechnology as tool for the crop to improve the quality of the crop. Today, we will talk in a topic of the origin of edible bananas and implication for banana breeding. Welcome, Dr. Hugo.
Okay, thank you for the introduction. Okay, does it show properly on your screens? Can you see the title slide?
Yes.
Okay, good. So I have been working in Thailand and Southeast Asian region for the past 25 years now on banana diversity, wild bananas, banana domestication, and so I have some things to say. I will talk briefly about, let's say, the standard model of banana domestication, which has been evolved since Simmons 50, 60 years ago introduced something similar into a standard model published by Perrier in 2011. I have several critical comments about it. So I will raise those. And I will then conclude that there is actually a different way the bananas originated and rather spontaneous model. And that has implications for how we do breeding for better cultivars. Okay, briefly, this consensus models are published by Perrier and colleagues in Proceedings of National Academy of Science in 2011. They base it on a multidisciplinary research. And what does that mean? It means there is genetic analysis in there. There is archaeological studies, and there is a part linked to linguistics. Essentially, what the model implies is there is a very tight linkage between the banana domestication and human interference. Initially, you have the wild species, and people were interested in those wild bananas already, not for the fruits, but for other purposes. That led them to take them along during human migrations, bring the different wild species in contact with other varieties. They made spontaneous crosses. From that derived then some progeny that had reduced fertility and maybe a little bit edible. Then further migrations and further crosses with wild species led ultimately to edible varieties diploid, and at some point two gametes were formed and they hybridized with a other source again to form triploid cultivars that we all refer now. So for the genetic analysis, this is the figure that is used in the Proceedings of National Academy of Science article, and this is their caption. This is based on microsatellite analysis of 22 loci in 41 wild accessions and 131 cultivars. And so, okay, what they claim is these are different cultivar groups shaded in gray here, and the colored lines indicate the different subspecies. And so they're named here: Microcarpa zebrina, Malaxensis, Cyanea birmanica, Banksii here. And what they notice is that many of these cultivars form clusters between Banksii at one end and Microcarpa zebrina at the other end. Keep in mind that this kind of representation, you can actually flip over the branches. So actually this Microcarpa zebrina branch you can put in here as well. It's exactly the same phylogenetic analysis. And then you clearly see that these are intermediates between zebrina and Banksii, while here pisang mas, kwekai, and fig bananas are then intermediates between zebrina and Malaxensis. But the main focus is here on these cultivars as intermediates between Banksii and zebrina or Microcarpa. And so you see a gradual range of more Banksii-like to less Banksii-like, more Microcarpa or zebrina-like, and also a range from clearly Papua New Guinea cultivars to some still Eastern Indonesia cultivars, and some strange occurrences of these cultivars. So some have been taken to other locations. So they see a decrease in Banksii contribution, increase in zebrina contribution, and that goes together with an increase in heterozygosity. And their major focus is here. Banksii is involved in all of this. They mention here also the cytoplasmic contribution, mitochondrial contribution, which is not depicted in this picture. And you don't have hybrid mitochondria, okay? There is no heterozygosity in the hybrid. In the mitochondria, there is no these are haploids, genomic parts. So there is no hybridity in there. So this is completely a wrong depiction of what is going on. But the key is here that they link it to Banksii and archaeology of Papua New Guinea, early agricultural settings, has indicated that in Papua, in the Musa Acuminata Banksii region, there are indications that people started to cultivate plants already 6,500 years before present, and maybe even 10,000 years before present. That's been published in a paper in Science, early 2000s. And so in a location in the center of Papua New Guinea country, so rather southern quarter of Papua Island, there is in the mountains a valley where there is a swampy area now, and so swamps and bogs are very good for conservation of pollen and other archaeological sources. So they did an analysis there, and there are indications in the soil that people actually did things there. There are indications of post holes, so where people dig holes to put in a piece of wood, maybe to build something, and there are indications of things going on. So people were there. They also find charcoal. So here, this graph here indicates the timeline from 2,500 years before present all the way to about 10,000 years before present. You see changes in the amount of charcoal, changes in the pollen composition that can be recovered. Where at some points there is more of a forest-like environment, some points there is more of an open grassland environment. And looking for phytoliths, they find phytoliths of Musaceae in the environment. So phytoliths are very small silica particles that plant cells make as part of their cell wall structure. It probably contributes to making plants inedible for certain herbivores, insects, and other small animals. So these silica structures, they are almost indestructible. So even from in soil samples that are apparently 10,000 years old, you can find these structures and you can identify them to some extent here even to the different species. So you have in these diamond shapes, this is phytoliths of Ensete. This square here is a phytolith of Musa ingens, the largest banana species that occurs in Papua Island. And so these black circles here are indicators of the presence of phytoliths from Musa species, so Musa acuminata banksia presumum. The graphs here indicate the number of phytoliths total found for grasses and for Musaceae. So combined the genetic analysis indicating a Musa acuminata banksia component to the cultivated bananas, Musa acuminata banksiae originating or growing predominantly in Papua Island, and here you find phytoliths of bananas in a cultivation setting 6,000 years ago, 10,000 years ago, 2,000 years ago. So there is some indication that people were doing something, they were probably growing crops, and it could have been Musa, could have been bananas. In addition, so they add the linguistic component, and linguists have analyzed the many languages spoken in Papua and Micronesia, Polynesia, Southeast Asia, and there is a clear indication of relationships between the languages, between the cultures associated with them, and maybe also with the bananas that those people grew. So by studying the words that people use for banana in a particular language, you can find similar words in related languages, and that seems to form a chain of evolution of the languages and migration pattern over the people speaking those languages. So these three elements combined in the opinion of Perrier and contributors make a strong case for bananas as cultivated plants to originate from Papua New Guinea area, from the Banksia area. And they claim that happened in a gradual process from fully seeded bananas to proto-cultivars with still a lot of seed, but some utility maybe as edible bananas, to primitive cultivars that are still fertile but produce limited seed, and then finally to completely edible forms without seed. So I have several questions about this issue. Do cultivated bananas really cluster with the subspecies Banksia? When you look at the phylogram that they present, it seems reasonable, but actually, is that really what it is? Then, if really people did agriculture 10,000 years, 6,000 years ago in Papua New Guinea, did they cultivate bananas? If yes, did they cultivate the bananas for the fruits or for other purposes? And I'm not thinking about leaf or for fiber or for shelter, because you can cultivate, you can get those from the wild plants. You don't need to cultivate bananas to get some leaves. But people may have been cultivated the bananas for the corn. Then I have several issues with the linguistic argument. I'm not a linguist, but from studies of human genetics and languages combined, it is very clear now that the evolution of languages and the evolution of human populations, they are not really congruent. For instance, in many Pacific islands, people speak an Austronesian language, but genetically, they're more related to Papuan people. How did that happen? So it's not really clear. There may be different stages of migration, which spreads the genes of people, and then culture is an additional layer which may have spread later on. So even if languages and people might spread nicely in a congruent pattern, did they necessarily take bananas all along? What I found out by talking to some local people on Sulawesi and also in Thailand and in other parts of Indonesia, people use the same word for the wild and cultivated bananas. It's unti biji in Sulawesi, and unti or punti is for wild or cultivated banana. So yes, there may be nice linguistic relationships, but that does not mean they are related to the cultivation of bananas. And then my main problem is whether this was really a gradual process. Just keep in mind, we have to end up with a seedless banana. And as we know now, it's very difficult to do breeding with a seedless banana. You don't have progeny. So how did the early farmers manage to solve that problem? You want to breed for anacarpy, for edibility, for low seed, but actually if you want to do breeding, you need seed. So my first point, it's not only Banksia, and not even mainly Banksia, I would say. Some arguments from other people, actually Edmond de Lange and Xavier Perrier and Tim Denham and others, the authors of the NASP publication, they already mention in another publication that there are separate, different origins for the bananas. They call it the banana split. The Pacific AAB bananas and the African AAB bananas have a very similar genetical background, but still different from each other and from different regions. And they never mixed. They never got, were never taken together by people. So in their original banana split publication in, where I forgot to, anthropology publication. So they mention here two separate parts of the region of origin of bananas. And they have two distinct domestication and hybridization processes. So this is more one of the pictures they show here. So here's Papua Island. So here is supposed to be the origin of bananas. And these AAB bananas, of course, you need the Balbisiana contribution. It's not clear whether Balbisiana is native to Papua. It has been found there in the forest. And I haven't been there to verify myself what is the condition. So I don't know. In this publication, they argue that Balbisiana was only native to northern India, southern China, very northern part of Peninsular Southeast Asia, so not in Thailand, but northern Burma. In some publications, they claim even not in the Philippines. Here they indicate that in part of the Philippines it may have been native. And so they propose that Balbisiana plants were introduced from South China or the Philippines to different regions of Peninsular Southeast Asia, one hand farther east to the Moluccas and Papua Island, on the other direction, more westerly through Borneo, Sulawesi. And because of these two separate migration patterns of people and bananas, they ended up coming in contact with the early AA proto-cultivars from Eastern Indonesia at different locations at different time points. And so they have that separate origins of the AAB hybrids from there. That's a nice story, and indeed, my analysis of haplotypes in the cultivars, both African plantains and in the Pacific plantains, indicates there are separate origins. And within the Pacific plantains, I can even find 3 different origins genetically. I'm not sure whether that corresponds to the 3 different morphological types, the Maya Maoli, Iholena, and Popo Ulu types that have been identified based on morphology, but it could be. What I also find is that Pisang Sungguh is related to both types and is not mentioned by the banana split here. There may be a triple origin. So, but there are different origins. Also another, okay, here, let me show you the haplotype analysis here. These are 5 African plantains, from the horn plantain, the false plantain, and so on. And all of them are genetically identical. And they are different from the Pacific plantains, which are in 3 different types here. So Popo Ulu, Uzakan, and Mairitu are identical to each other. Rukumam and Luba are each unique. And then you see here Pisang Sungguh, Again, a unique type of plantain. Xavier Perrier, you see him here, and the Biodiversity CERAT group. They also published another paper in Annals of Botany in 2016. Using DART whole genome profiling, they find very complex evolutionary taxonomic origins for the edible bananas. They enumerate 117 distinct multilocus genotypes for the diploid bananas and 78 distinct multilocus genotypes for the triploids, both triploid A, AAB, and ABB. So all included here. And they mentioned there was great confusion over the grouping of the ABB triploid bananas. So if the bananas really originated from Papua New Guinea, Banksiai, and then they diverged in almost 200 different hybridization events, selection events by the early farmers. Just think how difficult it would be to get just one cultivar from a wild type by humans selecting for lower seed frequency, and let alone get 200 different crosses to result in edible seedless bananas. Haplotype analysis that I've been doing in the lab confirms these multiple origins. I have no doubt about that. Here are some of the triploid A bananas. So the red form one cultivar group, Leyte Koinomsa form a second group, the Gros Michel and Cavendish bananas form another group. They are not identical, but Koi Hom Tong is identical to Gros Michel. So there are different groups of the triploid bananas that you can identify. And in orangey here are haplotypes that I could say are somewhat Banksiai related. We find them also in Banksiai, but we find them also in some bananas from outside Papua region. In red here are more or less Zebrina-like haplotypes and pinkish Malaxensis. Purplish here are Germanica Siamea haplotypes. And you see here Kai Pratabong, Kai Tong Luang, and Koi Tin Tao. These are Thai cultivars. They have a Bermanica contribution. And you see also some Banksia in there. I'm quite sure people did not bring Banksia bananas from Papua to Thailand and make crosses with seeded plants here. So these Banksia types are rather ancestral. So these are the original types that were all over the Musa acuminata germplasm. And some remnants you can find still in other locations outside of Papua. This is a similar picture for the diploid edible bananas, different clusters. And I would like to point out here Pisanjari buaya, Koi Nujora K. This one has a very unique composition with Zebrina malaxensis contribution, maybe Banksia, but certainly ancestral, and also here in yellow indicated Halabanensis contribution. About these ABB triploids, the great diversity and the confusion that the team from Biodiversity and CERAT presented. Well, it's not surprising to me when I do my haplotype analysis, I see exactly that. Pisang awak Wenamwa, that forms a nice cluster, yes. But all the other ABB bananas are very diverse. Not so much in the Acuminata contribution, which is quite consistent and either ancestral or rather Eastern Indonesian for most, or more Malaxensis debrina, Western Indonesia for some others. And Romanica siamea for the Thai cultivars. But there is a lot of diversity in the Balbisiana. Well, not as much as in Acuminata. In Acuminata, for these, you see here 58. For this particular locus, we find 95 different haplotypes. There are only 7 that I found so far for Balbisiana. So Balbisiana has only about 10% or less of the diversity of Acuminata, but still there is diversity. And it is the diversity in the Balbisiana that makes this ABB banana so diverse and confusing. This leads me to the question whether we need humans to be involved in the origin of the edible seedless bananas. And from my explorations in Thailand, I see that Musa acuminata can make interspecific hybrids spontaneously in the forest with all kinds of other species. We know with Musa balbisiana, it's in the edibles. Musa schizocarpa, a very few edible bananas are combinations of Acuminata schizocarpa. But I have now good whole genome proof that Musa acuminata has made hybridizations with Musa yunanensis in northern Thailand. And I have morphological proof that Musa acuminata makes hybrids with Musa rubra. So here is a stool of hybrids, most likely hybrids between Acuminata and Musa rubra. The two original species occur within 5 kilometers of this particular plant. At some other places, I found suspected hybrids and the original plants within 100 meters. These fruits are empty. Some seed develop, but it's empty, cannot germinate. So we have sterility, no parthenocarpy. Morphology is intermediate between Acuminata and rubra. These are hybrids between Musa acuminata and Musa yunanensis. I'll talk more about this story later, but these also are triploids, not diploids. I suspect these are diploids. The Musa acuminata rubra are probably AR genome. These are AYY genomes. So 2 yunanensis and 1 acuminata genome. That happens in the forest, naturally. Also in this case, the plants form seed, but I have not been able to germinate a seed. It's, I would say, 99% empty. I have not found many seeds that sink when you put them in water. They're almost all, as far as I can tell, exclusively 100%, but let me say 99% floaters, empty seed. So there is sterility, but not parthenocarpy. Musa acuminata not only forms hybrids with different species, it forms hybrid between the different subspecies in the wild. And that is indicated by another publication by the same Xavier Perrier is here, the same CIRAT Bioversity group. They did some SNP genotyping for here 14 wild banana accessions, 9 cultivated bananas, and a Musa balbisiana here. And so what they found are two important things here. Some of the wild here are hybrids between here Microcarpa and Zebrina, or here between Birmanica in yellow, Birmanica siamei in yellow, and Malaxensis in blue. And this is a much more complicated case here. This is a wild banana with contributions from Siamea birmanica, Malaxensis, but also Zebrina, and mind here, even Banksii, even Balbisiana. In the wild, you have all kinds of things going on. Cultivated bananas here, they look similar here to the Microcarpa wild banana. They have Zebrina and Banksii contributions, Banksii green, Zebrina red. You see the same kind of thing here. They also have something strange here. They call this a cryptic contributor. No surprise to me. Already in 2007, 2008, I discovered that many of the wild accessions from ITC are actually rather hybrid accessions. You have here a Tavoy type of the Birmanica, is actually a hybrid between Zebrina, Malaxensis, and Birmanica. I shouldn't talk about hybrids. These are admixed genotypes. Borneo, Microcarpa is, ancestral Banksii-like, but also has Zebrina, Malaxensis contribution. Also here in Thailand, this is the wild from Thailand, Siamea, but you find here Zebrina and Malaxensis admixture. Pahang, the model of Malaxensis type, there is some contribution by Zebrina. So no surprise to me. This is from an exploration in Sumatra, wild bananas that I did together with Fajar. Here are indicated the Zebrina malaxensis morphotypes, Sumatrana types, and Halabanensis types. And you see several of these bananas are admixed between here some Halabanensis contribution, some Zebrina malaxensis contribution. Here within the Halabanensis, you see some Sumatrana alleles popping up, or some Zebrina alleles. Within the Sumatrana, you see typically Sumatrana, but also Malaxensis or Halabanensis alleles popping up. And you see some haplotypes here, some alleles that honestly we have no clue where they come from. So it is rather confusing. It is rather admixed, naturally, not because people moving things around. It happens in nature. Talking about that cryptic contributor a little bit, so they found that a lot in Pisang Madu and some other cultivars. Well, there are in, for instance, Gros Michel and Cavendish bananas, there are some haplotypes that I cannot assign to any wild accession. So haplotype number 13 for catalase is nowhere to be found in ITC collection. Haplotype 9 at isocitrate hydrogenase is nowhere found to be in ITC collection. And so far, I have not found it in Thailand, and we have not found it in Sumatra. So there are haplotypes that are unique, but they're not cryptic to me. By looking at the sequence, I can tell that they are related to some others, and so they are clearly within the Zebrina malaxensis group. So 13 red here indicates Zebrina, the pinkish indicates malaxensis. To be honest, Zebrina and malaxensis at the haplotype level, at the sequence level, are very difficult to consistently distinguish. The morphology and the genetics there do not give a clear conclusion which type is what. But there's nothing cryptic about it. These are in the wild somewhere, but I know not in Thailand, and we know now not in Sumatra, not most likely not in Papua, because there's quite a lot of wild accessions from Papua in the ITC collection, and we don't find it there. So it's most likely somewhere else, Peninsular Malaysia or Borneo or Java. So where does that lead me to? First of all, there's the phenomenon that the Musa acuminata genome or karyotype is rather flexible. The very chromosomal rearrangements have been observed, described already by Ken Shepard's karyotyping work. There is also in 2016-2019 two more publications by the Biodiversity CIRAD group, Guillaume Martin and Frank-Christophe Borens. Okay, Xavier Perrier is not in the author list, but he's involved in this group too. And these chromosomal rearrangements that Dr. Hans de Jong will talk about next more, they seem to be somewhat restricted to subspecies and geographical areas. Not strictly, but you don't find them just anywhere. There are restrictions on where you find them, in what subspecies and what geographical location. And why did that happen? Well, that can be explained because of several barriers to gene flow during changes in sea level from the ice ages. So sometimes the islands of Southeast Asia were connected to each other and to the mainland. Sometimes they were separated when sea levels went up or down. And in addition to that, what I firsthand experienced from my exploration with Faja in Sumatra is that the mountains play a very big role too. I didn't realize that the mountains in Indonesia, Sumatra, but also the volcanoes on Java, they are really high, much higher than in Thailand. And so they create very distinct microenvironments on the mountain slopes where the different banana species can co-evolve with their pollinators and seed predators. And so that has led to a very mosaic pattern of occurrence of certain morphological types, but presumably also these different karyotypes with the chromosome rearrangements. Those different bananas, different morphology, different karyotypes, they can still intercross, okay? Musa acuminata can even cross with different species, as I've shown, as we know from the AAB and so hybrids, as I've shown from the AYY hybrids. So it can form hybrids even with different species, so why not with its other subspecies? When, however, two Musa acuminata genotypes with different chromosome formations cross, then things can go wrong at meiosis. And Dr. Hans will talk more about that. But the result is that when subspecies of different chromosome types intercross, most likely you get reduced fertility or complete sterility. So the AYY hybrids that I find in Thailand, they are 99, if not 100% sterile. When this sterility from these crosses occurs together with parthenocarpy, you have seedless fruits, edible fruits. And that could happen within just one cross. So it can be instantaneously. So what are the implications for breeding? Well, the Musa germplasm is very diverse, as everyone knows. Everyone agrees on that. Not all of it has been collected and characterized. And I think a very important part of it is still missing. What they call the cryptic species. And so what I know are some populations of Zebrina malaxensis, like wild bananas, is still missing in the international collections, and probably also still in the Indonesian collection in LIPI. It certainly is missing in the Thai collections. We have to find it. We have to locate it. The fact that you can have sterile fruits by just one crossing means that bananas, the edible bananas, are essentially completely unimproved. There has not been any selection for any trait at all, not by the early farmers, and as we know, not in historical times, not in the last 50 to 100 years. There has not been any thorough selection for any trait. The only trait that has been selected for is parthenocarpy. So the breeding with the wild forms for traits that are desirable for human consumption can be very fast if we identify the proper germplasm. And sterility can be achieved in one generation if you make a right cross, and probably it can be combined with parthenocarpy in one generation. If we study these traits a little bit more in detail, we probably can design a breeding scheme where we can achieve success in making completely new edible bananas. So how should we do that? First of all, first step is we have to do still a good job identifying the germplasm. Some germplasm is missing, and we have to identify the traits that we are looking for. I have found bananas in Thailand, in Kanchanaburi province, in Khon Kaen province, growing in, I would say, dry dipterocarp forest. If you want to find drought resistance, you have to go there. It is present in the wild. About disease resistance, as I said, I have never seen severe disease outbreaks in the wild bananas. So I don't know whether they are resistant or just lucky avoiding the diseases. I think it's rather the latter part, because of the environment, that the diseases never get a good foothold and never can spread far and wide. So we have to do still a good job with the germplasm identification, genotypically and especially phenotypically. We have to study the loci involved in parthenocarpy. There has been some studies done in the past. Simmons and colleagues also in Africa 10, 20 years ago, they did some studies, and they indicate 1 or 2 loci involved, or maybe 3. Recently, a GWAS study has been done, and again, we come up with 1 locus. I can guarantee you there are most likely many different loci that can give you parthenocarpy in the different backgrounds. So what we have to do is we have to identify the different, I would say, karyotypes. And within each of the karyotypes, we have to study for parthenocarpy. It happens in Malacensis background. It has been found on Sumatra. Also, during the Fusarium resistance mapping project, Fajar found one of the progeny apparently having parthenocarpic fruits. So we have to identify the different chromosome contributions, the chromosome configurations. We have to identify the different ways you can get parthenocarpy. And then, or at the same time, you can start making crosses within chromosome configurations to improve particular traits. So if you want to improve drought resistance, you can do that probably in the Siamea burmanica chromosome configuration subspecies. You can probably also do that in particular types of Malacensis zebrina from particular regions of Borneo or Java, or other places where there is a seasonal drought that's quite severe. You can make crosses for improved harvesting index, so bigger fruits at shorter rotations. You can do all kinds of improvement with the completely seeded wild bananas. And then at some point when you're happy with your product, you have to make a cross with an appropriate accession from another chromosome contribution that together will give you parthenocarpy in most likely one generation. So you make one cross and out come the products which you can then sow, and they will then be sterile and parthenocarpic. At least some of them. And we don't have any problem doing vegetative multiplication of the desirable genotypes once you have them identified. So we should focus on breeding within the wilds, improving the wild accessions, and at the same time study the karyotypes in Musa and study the different ways how to get parthenocarpy. And then you can produce hundreds, if not thousands, of sterile parthenocarpic progeny from various processes. So with that, I have to thank my employers, so Kasetsart University and Center for Agriculture and Biotechnology, and also some funding from AgBio Perdo project here. And I would like to thank you for your attention.
Thank you, Dr. Hugo. Anybody has questions?
Well, probably I can thank Hugo for his excellent presentation. What I like especially is your very clear indications what you can do on the level of plant breeding. I mean, a lot of people talk a little bit about it, but they never are precise what are the restrictions and what are the challenges. And you made great effort, and I think should publish that, Hugo, as soon as possible. Very nice indeed. Thank you.
Thank you. I'm working on that.
Hugo, can I ask a question for you?
Sure, go ahead.
Yes, it's very nice presentation and cannot wait for your publication for this topic, really. You mentioned that you found the triploid AYY. Can we indicate which one is the male parent and which one is the female parent, or you have idea how to identify which one is it? Hugo, you mute your microphone.
I have the chloroplast, mitochondria, and nuclear genome sequences. I have done the analysis. I can look it up for you which one is the chloroplast type, but I can tell you exactly what's the contributions. I don't know by heart now. I'm confused whether it's the acuminata or the I think it's the Unanensis chloroplast mitochondria. But
I think it's quite interesting because how the triploid is formed the triploid gametes of pollen or from ovary.
I think that it's a diploid Unanensis ovule. If indeed we did some study also together. We checked when we did the progeny of your Malaxensis cross, and we found the mitochondrial and chloroplast inheritance, and we confirmed that it's pollen contributed, right? Yes, so that's so I think it's diploid pollen on an acuminata ovule. The problem is that, well, we're still working on the final details and trying to figure out what's exactly going on. But I think originally there must have been a triploid formation from a diploid Unanensis with a diploid Unanensis pollen on acuminata ovule that formed a triploid hybrid progeny. And it's this triploid hybrid progeny that produces diploid Unanensis pollen. Through what we know flow cytometry, it's clear triploid. The haplotype analysis so far suggests it is clear triploid for all loci. The pollen analysis indicates there is something strange going on. Some of the accessions have mixed pollen with small and large grains. Others have consistently one type of pollen grains. I think there is something going on here with the meiosis in these hybrids. And we have to do some more study, but the thing is, I don't find many diploid Unanensis plants in the forest in Thailand. So far, we found 2 in one location. Over the whole area, several hundreds of square kilometers, where I have collected several samples, we found only 2 diploids.
Yeah, well, it's very interesting. Thank you.
It's strange.
We
It's
have one
certainly-
more question
Yes,
from
yes.
Mrs. Newhouse Saya.
Yeah, I see a hand raised. Go ahead.
Okay. You did open. Please turn on the microphone.
Hello, my voice is loud.
I
Thank
can
you.
hear you now.
Good day. I'm Nurhalisa. I would like to know about the Musa acuminata for Achan Hugo, because this one we have cultivar in South Sulawesi about banana is the Musa acuminata, and I don't know. This is the cultivar is already to know the haploid or not. We don't know, because in our university until now we don't have the like facility for now. What is the haploid from the banana? Okay, may you help me how to select or how to step by step for the assist for analyze the cultivar of our banana cultivar. Thank you, Achan Hugo.
Okay, so if I understand well, so you say you have cultivars, banana cultivars in South Sulawesi, and so I assume
Yes.
they are seedless. That's why I call them cultivars, right? And
Yes,
you want
that's
to,
right.
you want to know what kind of cultivars are these? Are they diploid, are
Yes.
they triploid, and so on, right?
Yes, right.
It's getting very easy how to do that now.
Mm.
I don't know how many you have, how many you want to analyze. And if you don't have many facilities, I don't know what facility, but first thing to do would be to do some flow cytometry, if you can do that. If you cannot do it there, I'm sure Fajar might be able to help you with that.
Yes, we have the fly
Okay,
cytometry
okay.
in our lab
You have a flow
Mr.
cytometry.
Fajar.
and still available.
So you could do it here, but I know there is concern with sending materials across borders. So if Fajar can do it, probably you can arrange with him. Then
Okay.
if you extract DNA, to be honest, I would say just send it for sequencing.
Yes. Okay.
There are, at the moment, the cost of sequencing is $100 for a whole genome. So you cannot do it. You cannot do it for hundreds of cultivars. You probably won't have the financial resources. But if you want to do it for 5 or 10, you probably can find some resources to do that. Okay. So
yes, thank you so much.
Yes, there are techniques you could do with microsatellites. You can do with rapids. You can do with, but all those answers they give are inconclusive. You then have to add reference cultivars from ITC or other places, and so it becomes a very complicated job. And since it is the cost of whole genome sequencing is now less than $100, just do it. You will have to send the DNA abroad, and
okay.
I'm not sure what are the regulations restricted, but just with the DNA that you isolated, no one can recreate a plant at the moment. Not in the next 10 to 50 years, people can recreate a plant from extracted DNA. So there should not be any fear. Should not be any fear of sending sensitive biodiversity material abroad, and that people will later on patent it. So there should not be any fear of sending DNA samples abroad. So you probably can coordinate with Fajar, and Fajar can coordinate with me. You can coordinate directly with me how to do it. But so for less than $100, you can get it done. If you have 5 or 10 accessions to sequence, that might be possible to do it. And then you have all the answers that you would like to have. Whether it's what is the level of heterozygosity, what is the relationship to other bananas. There are already several banana accessions available from NCBI, from public databases. So you don't have to do the sequencing yourself. You just download the results from GeneBank and you compare with them. If you send us your email, we can get in touch. For your information, end of May, we will organize a bioinformatics workshop here in Kanbangsaan in Thailand for 1 week. I can send you the information, and it's especially for people like you that we want to do this, people who are not sure how to do sequencing, how to analyze, and what to do. You come here. Thailand is opening up, so you come here for 1 week and you learn all the techniques and can work together on this. Fajar and me and you, we can, and there are some other people from Indonesia here. I see Leah is here. I don't know whether Nani is around. She registered, but I don't know whether she's actually logged in. So there are several people in Indonesia who can help you out, and we can coordinate and get it done together.
Thank you so much.
By the end of this year, you can have all the answers you want if you start working on it.
Thank you so much.
Thank you, Dr. Hugo.
Thank you, Mr. Fajar.
You're welcome. May I give a small remark to Icha?
Yes.
Icha, if you want to send a sample to me, it's very easy. So Green have new facilities in Cibinong. I think it's much easier. I think we can talk.
Okay. We talk more and deep later. Okay. Thank you, Mr. Fajar. Thank you, Achan Hugo.
Thank you, everyone.